So I will talk about 3 different topics today using examples from animals to illustrate how this framework for behavior can be used to understand how genes affect neurons, which affect brains, which affect behavior. And the 3 examples will be one example on the left side, moving from sensation to action in the innate responses to environmental cues. The second will be an example of internal motivation, using sleep and circadian rhythm as an example. And the third will address the question of why we don't all behave, where variation comes from that gives rise to different behaviors in different individuals. Now, in each of these 3 sections, I'm going to talk about one particular class of genes, not because it's the only class of genes that affects behavior, but because it is an important class of genes that affects behavior, and it serves to illustrate these principles very beautifully. And that class of genes are the genes that encode G-protein-coupled receptors. G-protein-coupled receptors are molecules that sit on the cell surface of neurons and also of other non-neuronal cells. They detect stimuli outside the cell and then propagate a signal to the inside of the cell that causes biochemical changes in the cell, classically the production of signal transduction second messengers like cyclic AMP or inositol trisphosphate. Now, I'm not going to talk about their signaling in detail. What I'm going to point out is that by transmitting signals from outside the cell to the inside of the cell, G-protein-coupled receptors are able to interface with many of the important kinds of systems that regulate behavior. Let's start on the left here with environmental stimuli. G-protein-coupled receptors on your sensory neurons are critical for the detection of odors, tastes, and light. When you smell a barbecue, or you taste a cookie, or you see someone walking across the room, the first event that happens in your nervous system is the activation of a G-protein-coupled receptor on an olfactory neuron, a taste cell, or a photoreceptor. In addition to their important role in detecting environmental stimuli, G-protein-coupled receptors inside the body detect signals that pass from one cell in the body to another cell in the body. These are internal signals. So, for example, adrenaline, the speedy heartbeat, and sweating that occurs when you're excited occurs because G-protein-coupled receptors recognize adrenaline that's been secreted into the bloodstream and tell different cell types that something has happened and cause them to change their behavior appropriately. Dopamine, a small molecule that's used to signal reward or the anticipation of reward and is implicated in important processes like drug addiction, also signals through G-protein-coupled receptors on neurons that detect this molecule. I'm going to start my talk today with talking about the response to external environmental stimuli, and particularly about the sense of smell. The sense of smell begins with G-protein-coupled receptors in the olfactory neurons of the nose. Large families of these receptors detect different chemicals in the environment. Different neurons express different receptors to detect different chemicals. And my favorite of all the G-protein-coupled odorant receptors is a molecule called odor 10, which detects diacetyl, a buttery smell. Odor 10 is a molecule that is found in the nematode worm Caenorhabditis elegans. C. elegans, as it is known to its friends, is an organism that's been very valuable for studying the nervous system as well as other biological processes because it's easy to grow in the lab and easy to manipulate at a genetic level. C. elegans, which is just a millimeter long, detects many different odors, and it responds to them showing innate preferences for different odors. So, for example, the smell of diacetyl is released by some of C. elegans' natural food sources. And therefore, when C. elegans smells this odor, it approaches it. It's attracted to the odor. Conversely, when C. elegans smells certain kinds of toxic odors, like nonanone, it's repelled by those odors and it goes away from them. Diacetyl is detected by odor 10, the G-protein-coupled receptor I mentioned before. The G-protein-coupled receptor is expressed in a pair of neurons in the tip of the worm's nose, these blue neurons here. They're exposed to the environment, and they detect diacetyl in the environment to initiate the odor preference behavior. Another pair of neurons right next to them in red detect repulsive odors. So you could think of odor 10 as a gene for a behavior. Odor 10 is certainly required for the worms to respond to diacetyl. So that's illustrated by comparing panels 1 and panel 2 here. A wild-type worm, or a wild-type group of worms, when placed in an environment with diacetyl at one location, will quickly approach the diacetyl and accumulate at the source of this delicious odor. If the odor 10 gene is absent, if there's no receptor, the animals now ignore the odor. They'll scatter at random across the environment as if nothing was happening. So that tells us that odor 10 is required for detection of the odor. But there's a more important question, and that is, how do you go from that question of detection to the process of attraction? How do you generate the behavior of approach? That could be because the receptor itself somehow drove the animal to approach the odor. It could be because the animal had learned about the odor. Or it could be because something about the interaction of the receptor and the neuron in which it was expressed was important. To test these different hypotheses, Emily Tremmel did an experiment in which she moved the odor 10 receptor from its normal location in the AWA neuron to an abnormal location in the red neuron, AWB, which normally detects repellents. So this was done using a combination of the mutant and transgenic technology, which enables us to manipulate the site of expression of any gene within this animal. She then asked whether these animals responded to diacetyl, and they did, but they responded inappropriately. They were repelled by diacetyl instead of being attracted. Moving the receptor to a cell involved in avoidance transformed the behavior from attraction to avoidance. And what this experiment tells you is that the neurons are what determine whether a response is attractive or repulsive, that the sensory neurons are encoding behavioral responses. They're starting the anatomical process of generating different innate responses. The blue AWA neuron encodes approach responses, and receptors expressed on this neuron will drive attraction. The red AWB neuron encodes avoidance responses, and receptors expressed on this neuron will drive repulsion. This example of an innate behavior comes from a very simple animal, but as we move up the evolutionary ladder, we see that similar principles apply in much more complex nervous systems, including mammalian nervous systems. For example, mammals like sweet foods or foods with amino acids that contain valuable nutrients. Mammals avoid toxins and alkaloids. We perceive them as bitter. And work from Charles Zucker, Nick Reba, and their colleagues has demonstrated that these mammalian taste responses are hardwired using exactly the same principle that hardwires worm attraction and avoidance responses. There are different kinds of taste cells in the tongue. T1R taste cells detect these attractive tastants. T2R taste cells detect bitter compounds. Each expresses certain G-protein coupled receptors that detect these molecules in your diet. Now, to ask whether the cells can drive these innate hardwire behaviors, the Zucker and Reba labs did the following experiment. They took a receptor called a RASL receptor. It's a receptor that's not found in nature. It's engineered in the laboratory, and it detects a compound that's not found in nature, that's only found in the laboratory. They put the RASL receptor either on the T2R cells that sense bitter compounds, or they put it on the T1R cells that sense sweet compounds, and they asked the mice, what do you think about this RASL ligand? Naive mice that have never seen a RASL ligand don't respond to it one way or another. But if they have this receptor on their bitter cells, they reject the RASL ligand. They perceive it as bitter. And if they have this receptor on their sweet cells, they drink the ligand. They perceive it as sweet. So the conclusion from this is that our basic acceptance of sweet and rejection of bitter substances is encoded by neural pathways. And this idea was intuited hundreds of years ago by the French philosopher and mathematician René Descartes, who in this illustration here showed that a person putting their foot near a fire would generate a nerve signal up an innate pathway that would cause them to remove their foot from the fire. They didn't have to think about this stimulus. They didn't have to learn about this stimulus. They had an anatomical pathway for an innate preference that told them that this fire was too hot. And so by understanding genes, neurons, and brains, we can see the beginning of a principle for one idea about how behaviors emerge, and particularly how innate behaviors are built into a nervous system to help an animal respond sensibly to its environment from its moment of birth.